Alright, hello everyone. I'm going to start the Lab 6 overview. This focuses on membrane transport, diffusion, and osmosis. So as it says in the lab, you want to make sure you review Chapter 5, Part 1, membrane transport, go over diffusion and osmosis, and the definitions I gave you for diffusion and osmosis in lecture are the same definitions I'm going to want you to know for to help you with the lab report, okay? So membrane transport, okay, we talked about in lecture about how there's two types of membrane transport. But first, what is membrane transport? It's a transport of substances across the membrane, a cell membrane, um, into and out of a cell. And the substances that are being transported across the cell membrane are being exchanged between the ICF and the ECF. And if you recall, the ICF is intracellular fluid, also known as the cytosol of the cell. ECF is extracellular fluid, which is a solution outside of the cell. So both ICF and ECF are solutions. So you have to remember, what are the two components that make up a solution? A solvent plus the dissolved solids. Okay. Now, in today's lab, you're going to use two different substances to represent a cell membrane. Keep in mind, a cell membrane is actually described functionally as semi-permeable. Okay, some solids can cross the cell membrane, all right, but not all solids. However, assume water can always cross the membrane. Now, if you look here, what we focus on are passive processes of membrane transport for this lab. Okay, the main passive process for membrane transport is called diffusion. Make sure you know this definition I gave you here for diffusion. Okay, diffusion focuses on the movement of solids from a high concentration to a low concentration of solids until evenly distributed in a solution. What do we mean by evenly distributed equilibrium? This is the same example I gave you in lecture, so this is serving as a review. Now, the difference between two concentrations of solids is called the concentration gradient. So here they use a sugar cube. When they drop it in the beaker of water, they've established a high concentration of solute. What will happen is this solute, okay, will begin to diffuse. In other words, the water's dissolving it. But we say focus on the sugar molecules. The molecules begin to diffuse. The sugar starts to diffuse from an area of high concentration, as it says up here, in the direction of an area of low concentration, which it tells you up here in the definition of solids. And these molecules of sugar will continue to diffuse from an area of high concentration to low concentration. Okay, where we have that area of high to low concentration in these two beakers here, that represents the concentration gradient. So you can see there's a concentration gradient here, and then the concentration gradient is a little wider here. The concentration gradient is where we have an area of high solid concentration, and as we move away, the solid concentration gets lower and lower. So an area of high next to an area of low solid concentration. As long as we have that, as they have here in these two, okay, diffusion will want to occur. And diffusion will occur until the solids are evenly distributed. In other words, until the solute, in this case, the sugar molecules, are evenly mixed with the water molecules. Well, how do we know? They turn the sugar molecules purple. So you can see here that the entire solution is uniformly purple. There are no areas that are a darker purple. The color purple is evenly distributed throughout the solution. That lets you know that the sugar molecules are evenly distributed with the water molecules, or thus we are at equilibrium. If you were to look at the concentration of sugar here and compare it to the concentration of sugar here, you would see they're equal. However, in today's lab, as I said, you're going to use two substances to represent a semi-permeable membrane. One is a seaweed extract called agarose. And when you take agarose in a powder form, is the way they sell it to us in labs, and you take that powdered form of the seaweed extract called agarose, and you dissolve it into a buffer solution that's been heated above boiling temp, and then you allow that agarose solution, okay, to cool, it will solidify into a gel that has pores in it. So that agar, agarose gel, functions as the semipermeable membrane. But in this lab, you're going to use another material called dialysis tubing. That dialysis tubing, normally when you look at it, it's rectangular in shape and it looks solid. But in actuality, the plastic-like material used to make the dialysis tubing has tiny microscopic or submicroscopic pores. So it's also a semi-permeable material. So diffusion normally, so in the lab, when you measure if diffusion is occurring, you're actually going to do it in the presence of a semi-permeable membrane, and it's going to be part one of your lab, okay, the first lab experiment. And the agar or agarose media is going to be functioning as your semi-permeable membrane, okay? What's going to differ are the molecular, sorry, let me do this. What's going to differ are the molecular masses of two dyes that you're going to be using in this first part of the experiment, part one, diffusion. Now, diffusion, okay, in the presence of a semi-permeable membrane, if we want to know if diffusion is going to occur, we first want to look for a concentration gradient. We want to look for an area where there's a high solute concentration next to an area where there's low or no solute concentration. Well, you can see this right here represents a membrane, and on one side we have a high solute concentration made up of these molecules of dye. Over here we have no solute, so technically speaking, we have a concentration gradient between these two solutions across the membrane. Okay, so diffusion wants to occur. The solids want to diffuse from a high concentration to a low concentration until we reach equilibrium. However, if a semi-permeable membrane is present, you have to ask yourself a second question. Is that membrane, is that semi-permeable membrane permeable to the solute? In other words, will the membrane allow the solute to pass through it? In our body, okay, not all solids can cross the semi-permeable membrane, but water always can. But in this example here, they show you not only is there a concentration gradient, an area of high concentration next to an area of low concentration of solute or the dye, but they show you the pores in the membrane, the holes in the membrane are large enough to allow the solute to diffuse in a direction from a high to a, sorry, I'm trying to, from a direction from a high to a low concentration. It's not showing up right now. And these molecules of dye will continue to diffuse from a high to a low concentration until we are at equilibrium, until we have an equal concentration of solids on both sides. Now, there are factors that can affect the rate of diffusion, and that's what this is talking about down here. Factors that can affect the rates of diffusion. Well, it says direct proportions. In other words, temperature. As you increase the temperature, it actually increases the rate of diffusion of solids. So they're proportional to each other. Increase temp, you increase diffusion rate of a solid from a high to a low concentration. Likewise, the concentration gradient, the greater the difference in the concentration gradient. So where it says high concentration gradients, they're saying whenever we have a greater difference in concentration between two solids across a membrane, the greater the rate of diffusion. The greater the rate of solids that will move across the membrane and how fast they'll move will be higher. So diffusion rate is higher. However, inverse proportions has to do with what are factors that can slow the rate of diffusion down. Well, one is molecular mass or size. In part one, you're going to be given two dyes. One's called potassium permanganate and one's called methylene blue. Potassium permanganate has a smaller molecular mass than methylene blue. Therefore, potassium permanganate has an easier time moving through the agar media, moving through that semi-permeable membrane than methylene blue, which has a larger molecular mass. So what do we mean by inverse proportional? The higher the molecular mass of your solids, the slower the diffusion rate. Okay? The lower the molecular mass of a solid, the higher the diffusion rate. So proportional, higher, slower, or lower, faster. Okay? Media density. Okay? Basically what that means is sometimes what we do in our labs, we do it for our majors, is that we give them these Petri dishes with this agar gel. Okay, that media. It looks like a gel, but like I said, it functions as a semi-permeable membrane. It has pores in it. But what we'll do is we'll use different percentages of agarose or agar to make the gel. We'll give the students one plate that'll be, say, 2% agarose or agar media, and we'll give them another plate that's 4% agar or agar media. The difference is that we use twice as much powdered agar or agarose to make the 4% gel than we did the 2% gel. Therefore, the pores in the gel, which is functioning as our membrane, are smaller. So here it says media density. The higher density of the media, in other words, the more agar or agarose we use to make the gel, okay, media, the smaller the pores, therefore the molecules have harder time moving through those smaller pores, so it slows down the diffusion rate. So in other words, what they mean by higher density is the greater the amount of agar we use, the more dense the gel is, the less permeable the membrane is, and the slower diffusion rate. However, the less agar we use to make the agarose we use to make the agar gel, the larger the pores are in the membrane, and therefore the faster the diffusion rate. So as I said in today's lab, it'll show you some pictures, and you're going to see it'll show you a regular figure, then it'll actually show you a picture of an agarose gel with these two dyes, methylene blue and potassium permanganate in there. Okay, and what's going to happen is both dyes are going to be added to one petri dish, so you're going to be looking down onto the surface of the gel where the two dyes have been added. And what's happened is we've taken a straw and we pushed it into the gel to remove a little plug of this agar gel. The gel is about a centimeter thick. So you're looking down at the gel, and then what you do is to the holes you put in the agar gel or agarose gel, you add methylene blue to one, and you add potassium permanganate, which usually has a purple color. It doesn't look purple in the lab, but it usually has a purple color, to the other one. If you look, it says the formula weights. Well, the formula weights are referring to the molecular masses. So potassium permanganate has a formula weight or molecular mass of 158 grams per mole. Methylene blue has a formula weight or molecular mass of 374 grams per mole. Therefore, the dyes of molecule that make up the methylene blue are much larger than the dyes of molecule that make up the potassium permanganate. Okay? The way it works is when you add the drops of dye into the holes in the gel that you placed, automatically you've created a concentration gradient. Okay? And the difference in the dyes, molecular mass. Okay? What's not going to change is that they're both added to the same agarose gel medium. Okay, same percentage, so the density of the gel is the same for both dyes. And we place them on the bench at room temperature, so the temperature is the same for both dyes. So what is constant in this experiment is the molecular density of the agarose gel medium. Okay, it's the same for both dyes because they're added to the same gel medium on the same plate. What else is the same is the temperature. Okay, that you would have placed in person, you would have put the plates at room temp, which is roughly 22 degrees Celsius. For both dyes, they're on the same plate. So the only determining factors that affect the rate of diffusion, okay, in the end, is going to be the molecular masses. In other words, their formula weights. The higher the formula weight, the larger the molecules of dye. The lower the formula weight or molecular mass, the smaller the molecules of dye. Okay. And it says here determining diffusion rate. How would they determine diffusion rate in the lab? I've get posted for you the lab protocol. You'll see they have a ruler and they're measuring the diameter of the dyes. So what'll happen is they'll tell you after 60 minutes, they'll measure the diameter of the dyes. They'll take a ruler and measure the distance that the dyes have diffused, or in lay terms, measure the distance that the dyes have spread out. Okay? Which dye can diffuse from a direction of a high concentration to a lower depends on which one has a smaller molecular mass. Therefore, it has an easier time getting through the pores in the gel. And I forgot one other thing. They're going to ask you a couple of questions about dependent and independent variables. Okay? Listen carefully. In science, whenever we talk about a term called independent variable, the independent variable is what you choose to use. What is the percentage of agarose used to make the agarose gel medium? What dyes did you choose? Those are independent variables. The dependent variable is this right here. The dependent variable, what we call the dependent variable, the reason why they say dependent is because it's dependent on what you chose for the independent variable. So depending on the percentage of agarose you use to create the agarose gel medium, and then depending on the molecular masses of the dyes, those are your independent variables. then those variables here, okay, affect the diffusion rate. So another way to say it is the diffusion rate is dependent on what's your percentage of agarose gel and what are the molecular masses or formula weights of your dyes. So the diffusion rate is dependent on these two factors. So again, this is your dependent variable. These are your independent variables. Part 2 is going to focus on osmosis. Just like for the lecture, I want you to know the same definition for the lab. Focuses on the movement of the solvent water across a semi-permeable membrane in the direction of the higher solute concentration until equilibrium. Assume that the semi-permeable membrane always allows water to move or diffuse across it, but only some solids. That's what this example is showing you. They take a region, they show you two solutions. They are sugar solutions. Okay, but over here we have a higher concentration of solute or sugar here, and over here we have a lower concentration of solute. They are separated by a selectively or semi-permeable membrane. Okay, the sugar molecules want to diffuse. There's a higher concentration here than here. So the sugar molecules want to diffuse from a high concentration to a low, but if you look here where they take a piece of the membrane in the two solutions and magnify them, notice that the sugar molecules here are too large to move through the pores in this membrane here. So they want to diffuse because there is a concentration gradient, but they're too large. In other words, the membrane is not permeable to the sugar. So diffusion can occur. However, osmosis can occur. We always assume the semi-permeable or selectively permeable, one and the same, and I said that in lecture, one and the same. We always assume water can move across, and that's what they're showing you here. Water is able to move across the membrane. Now, water's moving both ways, but there's a net movement of water. Notice most of the arrows are going from our left to our right. So what is happening? Follow the definition up here. Osmosis focuses on the movement of water across a semi-permeable membrane in the direction of the higher solid concentration. So notice 3 arrows are pointing from our left to our right. Only one is pointing the opposite way. So the net movement of water is across the semi-permeable membrane towards the higher concentration of sugar or solute. Water will continue to diffuse towards the higher concentration of solute till we reach equilibrium. Well, equilibrium is the same thing as saying same concentration. Okay? Watch the lecture video. I explain how these two solutions do have the same concentration, and it doesn't have to do with total volume. But the catch is this: the greater the difference in solute concentration between two solutions on the opposite sides of a semi-permeable membrane, okay, the greater the amount of water that will move across the membrane, creating a type of pressure called osmotic pressure. Now listen carefully. Under part 2, they give you a web link for Pearson, and it tells you when you click on that web link, choose, it'll say lab 1. We're in lab 6, but on the web link it says lab 1 and it says diffusion and osmosis, and it's got a little tutorial for you to look at. Okay, you can just kind of pick and choose what relates to the lab. It's just a tutorial. You can actually do part 2 osmosis without that tutorial, but it does have some information that may help you out. However, it only has partial definitions for like diffusion and osmosis, so make sure you use the ones in the PowerPoint. They do have a couple of activities to test your knowledge, so I do recommend taking a look at it to help you out. Okay. Then what the lab's going to do is it's going to go into a part having to do with tonicity. Tonicity has to do with how the movement of water across the semi-permeable, how the movement of water across the cell membrane can create a type of stress or pressure called osmotic pressure, and may change the shape of a cell. Okay, so in other words, tonicity has to do with osmosis and water balance. Okay, the way it works in the lab, okay, is there's going to be a solution that you're going to place in the beaker. It tells you, hey, this solution is in the beaker. The solution in the beaker is your ECF. Okay, in other words, it's a solution that's surrounding the cell and exerting pressure or osmotic pressure on cells. Okay, now the solution that's in the dialysis bag, and that dialysis bag, it's the one I told you that's made up of a plastic-like material that has small pores that does allow water to move across. Okay, what you put in the dialysis bag, what is placed in the dialysis bag, that is your ICF, your intracellular fluid. Okay, and it says this is because solutions can have different concentrations of solids compared to solid concentrations within the cell. So what's in the beaker is your ECF, what's in the dialysis bag is your ICF. Now, depending on whether the ECF has the same solid concentration as the ICF, or whether the dialysis bag has a lower concentration of solids than what's in the ICF, or whether the ECF has a higher solid concentration versus what's in the dialysis bag or ICF, depending on what the concentration of ECF is versus ICF, we can call the ECF or the solution in the beaker either an isotonic solution, hypotonic solution, or hypertonic solution. So in the lab, again, in the lab, the solution that's placed in the beaker, that is your ECF. And then depending on whether it causes the dialysis bag to gain water, excess water, or to lose excess water, or to not gain or lose excess water. In other words, it maintains the ideal water balance, whereas the dialysis bag may gain too much water or lose too much water, where in this case there isn't water balance, we've lost it. Depending on what happens with the movement of water, the ECF is said to either behave as an isotonic, hypotonic, or hypertonic solution. You can see these three here. Again, I went over this in lecture, so go back and watch chapter 5, part 1. Look at the go over the osmosis portion to help you with the lab. So these are some beakers that tell you a few things, but I'm going to jump over here. The way we experimentally determine if osmosis is occurring is by weighing the dialysis bag. The bag you have filled with a solution, that the solution functions as the ICF, okay? Because the way it works is the dialysis bag is comparable to these red blood cells, okay? If you have a solution in the dialysis bag that is your ICF, and the solid concentration in the dialysis bag, your ICF, is equal to the solid concentration in the beaker, which is your ECF, then the ECF is behaving as isotonic. And the dialysis bag, when you weigh it once before you place it in the beaker solution, or ECF, and then you weigh it an hour later after you take it back out of the beaker solution or your ECF, if the bag hasn't gained any weight or lost any weight, then osmosis didn't occur because the ECF was behaving as isotonic. However, if you get a change in the weight of your bag, then that either means the dialysis bag has gained water because the beaker solution, which is your ECF, behaved as isotonic, or if your dialysis bag has lost weight, then that means the beaker solution, which is your ECF, behaved as hypertonic. Then that means osmosis occurred. We had a net movement of water in or out of your dialysis bag comparable to these red blood cells. And that'll let you know how is the solution in the beaker behaving in terms of tonicity as one of these three. So so you would measure the weight of the bag twice. So this is a table that's given in your lab, okay? This is a table here that's under part 2. Beaker solution here. Remember, that beaker solution is your ECF. Okay. Dialysis bag. The solution you put in your dialysis bag, that is going to be your ICF. So the dialysis bag with the solution in it, you can think of being your cell. Okay. Notice they say initial weight of the bag. If we were meeting in person, you would take out this dialysis tubing, which is flat. You would open it up, clamp one end. To the other end, you would add one of these 4 different solutions at about 5 to 10 mils. It's usually 5. Then you would clamp the other end of the bag. You would weigh the bag initially in grams. Then you would take each bag and put it in the appropriate solution. Notice here's the solutions you put in the beaker, 10% glucose, and all 4 beakers would have 10% glucose. Okay. Once you place the dialysis bags with their solutions, after you've weighed them in the beakers, you would wait 1 hour, take them out, dry the dialysis bags off, and then weigh them again a second time. So the time between these two weighings is 1 hour. Okay, so the time between these two is you'll weigh the bag once, you put it in the solution, wait an hour, and you weigh the bag a second time. And it tells you this in the protocol, okay? Then it asks you to calculate the difference in, if there's any difference in weight change. And then it asks you to convert that to a percentage, and it gives you the formula right beneath the table on how to convert that weight change to a percentage, okay? You can see this dialysis bags here, there really wasn't a significant change. It's very small. You can see when you look at the dialysis bag here placed in the beaker, dialysis bag gained a lot of weight. If you look at this here, you can see there's even a greater difference in solute concentration. So the bags gain even, looks like you gained even more weight. And then if you look here, notice the ICF in the dialysis bag, solute concentration is lower than what's in the beaker. So notice the bag lost weight. So you got to compare what you're looking at here. And how would that compare to slide 6 here? Okay. Listen carefully, everybody. When you do your labs, you have to study these labs. Okay, I was grading the carbohydrate labs, and it was real obvious there was a few people that are studying the labs, but not everybody. Okay, you have to study these labs, and I believe I even I thought I put it in the carbohydrate, but even if I didn't, the information was there. You need to look for the answers. I've said this many times to this class, and I know I've said it. You can't just put answers down. Lab is normally 2 hours and 40 minutes. That's just for the experiment. That's not even for the lab report write-up. So lab itself takes a lot longer than 2 hours and 40 minutes total to do the experiment, write up the report, including the answers, the correct answers, and then submit it. So we have a lot of people that there's an issue of comprehension. Focus, I don't know what it is. Okay, but that's on you. You have to figure that out. Okay, because there were answers that were the last 12 questions on the carbohydrate lab. I mean, that was in the PowerPoint. That was on the first page of the lab protocol. Should have gotten it right. It was even in bold type. Okay, so that's on y'all to go through all that. So once you finish filling out all the answers in the table, okay, you'll do the same thing. You'll submit an electronic copy. Now, what I want to bring up, I'm sorry, hold on. All right, sorry, everybody. I had a little interruption here. I'm going to finish this up real quick here. Okay, so this is your lab handout, lab protocol. Make sure you read through it carefully. This information on the first page I've given to you. Excuse me if you hear a little background. This is part 1, the first experiment, focusing on diffusion. It gives you a little diagram, but here's the actual agarose with the dyes, and they're measuring them for you, and they give you the measurements. So you just have to look at those measurements, look at what I gave you in the PowerPoint and what they give you in the beginning to help you with the answers. This is where it talks about dependent and independent variables, which I've already discussed. Part 2 is osmosis. It tells you to review. It gives you then the experiment. Here's that link to take you through a practice run. You can actually do the lab without it, but I recommend you take a look at it to help you because it's got a few activities to ask you questions. Then you'll go into the lab, and here's the table that I showed you already on the PowerPoint. Here's the table again. Here's that formula to convert the weight change, the difference in weight change that you calculate into a percentage given here. And then you're asked a few questions. And if you look at these questions for 3, 4, 5, and 6, it's asking you about the beaker solution. In other words, tonicity. Okay, the solution in beakers 1, 2, 3, 4, and 4, you're basically going to answer, is the solution in the beaker behaving as isotonic, hypotonic, are hypotonic or hypertonic, okay? All right, everybody. Y'all take care. That's the end of the recording.